Okay, so synthetic biology. This announcement was made by Craig Venter, which was, a kind of proof of concept, which is he took a existing cell wall and created a synthetic copy, basically, of something that already exists in nature, a bacteria that exists in nature, at the minimal level of the genome. Synthetic means taking sugars, which represent the different oligonucleotides, typing out the sequence on a synthesizer, a chemical synthesizer, stringing together piece by piece what represented something that looks almost exactly like an organism in nature. And after gluing them together using different yeast to glue this long strand together, injected it into a hollow cell wall. That cell booted up, it self-replicated, and there was a colony of these bacteria that grew and that are continuing to grow. And this was a pretty major breakthrough for synthetic biology because even though it is very similar to an existing organism, not some totally novel organism that exists, as a proof of concept it shows we can chemically synthesize self-replicating organisms as opposed to simply tinkering with things that already exist. And the potential for what that means is quite vast. It's a different type of biology. It's genetic engineering together with biology, thinking about it from an engineering perspective rather than recombinant DNA, which is trying to manipulate existing organisms, which is quite limited by the existing organism itself. There was a pretty immediate public reaction to this, which were things like, is this really synthetic life? People doubted the meaning of the scientific progress itself. Was it really that meaningful? There were questions like, was life really created in a test tube? Does it disprove biblical creation? There were covers of an unbelievable number of magazines and mainstream media that showed things like Adam pointing to a cell and saying, and man created life. And this one quote here, which is the scientists were accused of playing God after creating artificial life by making designer microbes from scratch. Could this wipe out humanity? And there were a lot of questions that immediately came up, like, what are the implications for the environment? What does it mean if you release an organism that is self-replicating into the environment? What does it look like for evolution? Is there any way to contain these organisms? If you've watched any science fiction movies, you could imagine the kind of doomsday scenarios that people were immediately thinking about how the environment and ecosystems were going to totally change. But there was also a lot of promise that people saw in this as well. So the ability to synthesize drugs more quickly and more efficiently, or bioremediation, having a major oil spill and being able to release an organism into the ocean that actually eats up the oil and fixes it, or biofuels. So being able to create fuels through organisms that express different biofuels instead. Really exciting stuff could come from synthetic biology, but we were asked to consider the risks and benefits of synthetic biology and to develop a framework for how to think about the different risks and benefits that might arise from synthetic biology. And the biggest risks that we identified were issues around potentially bioterrorism. So being able to, one example is if you look right now on the internet, you can find many different types of pathogens where the genetic code has been printed onto the internet, right? So smallpox, which we've eradicated, right, is now, you can actually go find the sequence for smallpox just by doing a Google search. Or yellow fever, or pathogens that we have long since eradicated could be recreated and synthesized. And there are people who are do-it-yourselfers, people who in their garage are creating synthetic organisms. Now it turns out it's hugely expensive right now to actually make something along the lines of what Craig Venter made. It took billions of dollars to make what he made. And so the idea that a do-it-yourselfer could actually create some sort of organism in their garage and release it into the environment isn't all that plausible today, but maybe in the future. And from an environmental perspective, thinking about biosafety measures, this was another big concern, which is a live organism that you put into the environment evolves and changes. And if you build in containment measures, like you build in a suicide gene or you restrict the nutrients that it actually has available, which ones survive? Well, the ones that can bypass those types of limitations that you put in. These are organisms that aren't predictable, right? So they change and reproduce, and each new generation gets stronger and better at circumventing whatever the different stopgap measures are that you put in. And so what we were asked to do was to look at this technology, which is really in its infancy, and to come up with some sort of framework by which to address the potential risks and benefits from it. So we were asked to review the developing field of synthetic biology, consider the medical, environmental, security, and other benefits, as well as the potential health, security, or other risks, to identify appropriate ethical boundaries to maximize public benefits and minimize public risks. And this was all in the letter that was provided to us by the president, which was, please focus specifically on these. And welcome Steve Hauser, who is joining us. And I'm just going through Syn Bio, so please join us up here. Yeah.
Hi, everybody.
We've already introduced you, so, okay. So I'm doing Syn Bio and thought I would hand it over to you to do Guatemala and the ethically impossible report. So we submitted this report to President Obama on December 15, 2010. You can have access to it by going to www.bioethics.gov. It's called New Directions, the Ethics of Synthetic Biology and Emerging Technologies. And what we did was we developed principles and 18 recommendations within those principles to try to say, not just how should you think about synthetic biology, but how much you think about addressing the ethical considerations in any kind of emerging technology. When you have the opportunity to assess it at this stage, right, not some stage where it's fully developed, where the risk had been fully realized, but you can actually think about it at this stage. And we developed these principles: public beneficence, responsible stewardship, intellectual freedom and responsibility, democratic deliberation, justice, and fairness. And within each of these, we have recommendations for what that means. So public beneficence, thinking about maximizing public benefits while minimizing the risk to the public, recognizing that there shouldn't be any single group of individuals who unfairly bears the risk of scientific research. Or responsible stewardship, recognizing that it's future generations and the environment for future generations that we are also trying to safeguard. So how can you be a responsible steward of scientific progress and research? Intellectual freedom and responsibility, this is an area where the intellectual property rights, for example, haven't been well defined. How do you foster freedom and responsibility in a kind of environment where, for example, you could try to block people from having access to the most basic building blocks of organic materials, of organisms? How do you foster an environment where there really is a balance between individual freedom and responsibility, but also the progress of science? Democratic deliberation, thinking about issues with respect to involving the public. People seem pretty unconcerned from a playing God perspective at the single cell organism level. But what about when we get to multicellular organisms? Or if you can actually create complex organisms? We should have an ongoing dialogue about what that means. And justice and fairness, thinking about how to distribute the benefits of scientific research and how to also distribute the risk of scientific research so that it fairly reaches those individuals who it most is designed to benefit and reaches people more generally. With all of this, we developed a principle that's sort of an umbrella principle called prudent vigilance. And prudent vigilance means there's kind of two ways in which you can think about scientific progress. You can say you should wait until all of the risk of science are known before you allow scientific progress to proceed. Or you can say, don't worry about the risks, just let it rip, right? Let science progress. It will progress anyway in other countries, so you might as well allow it to go forward. We developed a principle called prudent vigilance, which is to think about things as a balance between those two extremes, right? So carefully, don't over-regulate, and don't over-regulate before a technology has really developed. But have checkpoints along the way so that as a technology develops, you ensure that the regulation is keeping up with whatever's happening in the scientific field. The report was received as people sort of recognized the vigilance concerns of it, that we urged caution, that we called for enhanced federal oversight. In general, the reports received some media attention, and the White House seems to have responded by recognizing and appreciating the work. There seems to be some progress happening in the different agencies to whom we directed our specific recommendations, and we set an 18-month time frame after which we hope to follow up and receive reports back from the different agencies to whom we directed particular recommendations to understand where things stand. And I'm going to turn it to Steve to talk about human subjects protection.
Outstanding. Hello again, everybody. First, I want to thank Nita for that wonderful introduction.
We introduced you in your absence, Steve.
15 minutes with video. It was touching, charming.
Did you have the piece with my mother there? That's the one I wanted.
There was a video. It was
Outstanding.
lovely, yeah.
Well, I'm so pleased to be here, even though my timeliness didn't necessarily reflect that. So I apologize for being late. And I'm happy to take the baton for the next part of this. How many of you have heard about the Guatemalan events? So many have. And in some detail or in a little bit of detail? So, of course. But let me review this with you. I would start by saying, as you might have heard the introduction, you spoke about the nature of our group.
I did. I did a
Yes.
commission overview, yeah.
Yeah, so we are a pretty diverse group, and compared with earlier commissions, we are not uniformly steeped in bioethics, law, and the traditional boundaries of prior bioethics commissions. And in fact, we are very multidisciplinary, which makes things fun. We are, as you know, representing law, medicine, bioethics, nursing, engineering, basic emerging science. And we're a much smaller group. Since there are only 13 of us, we can actually get to know each other very well. And I come into this as an internist, a neurologist, a geneticist, but not a bioethicist, although someone who every day really has bioethical issues on our table that are that we may not think of as bioethical. We think of them as medical decision-making. But so for me, I would tell you that the flavor of this presentation will be a non-professional bioethicist, but someone who is learning a lot over the past year and a half by being close to people like Nita.
Can I ask a quick question?
Sure.
Is there staff behind you, or is it
So we are a group of 13 with a magnificent large staff. And I would say that I've never seen a staff make commissioners look as good as our staff does for us. In terms, as you'll see here, when we speak about Guatemala, the deep dives into the research that they do, and also into their ability to put pen to paper in a very sophisticated and fluent way. These are remarkable people. Many of them are young people, recent law school graduates, people headed into politics, academia, law. It's really a great group. It
We have, I
restores
think,
your
almost
faith
24
in
people on
government.
the staff right now. So we have the executive director, who I mentioned, Valerie Bonham, and she is the executive director overseeing the staff.
So,
There too.
there we are. So this all